Okay. I begin by thanking the physics department of NIT Calicut to invite me and to giving a talk here. So I have decided to talk about, this is the title of my talk, Bacterial Chemotaxis in Time Varying Environment. I have prepared some slides, but it is not at all important that I go through all of them. So I urge you to interrupt me, ask me questions if anything is unclear. As I said, it doesn't absolutely matter that I cover all the material, but even if we cover only part of the talk, there should be some take-home message. So feel free to interrupt me for as frequently as you want. Okay. I was told that most of the people who will be attending are from engineering physics background. Is that correct? Okay, I assume that is so. So I have prepared something. So if that doesn't match your level, I have assumed certain background knowledge. But if something is unclear, please interrupt me. So, and by the way, the talk is for 1 hour duration, right? So so it is talk plus question?
Am I audible?
The total time is 1 hour? Okay. So then please give me some warning, let's say 10 minutes before, okay? Around 7:50 or something, you can give me some warning that you have only 10 minutes left. Then I'll try to wrap up quickly. So this is the title of my talk, Bacterial Chemotaxis in Time Varying Environment. And most of these results are obtained, that I will talk about, are obtained by my PhD students. Shobhon was a PhD student in my group. He has now finished and doing postdoc at Taiwan. And Ramesh is one student who is currently working with me. So first, let us see. Since most of you are from physics background, I have not assumed much for your biology knowledge. So I will try to explain whatever little biology we need to know to follow this talk. I will try to explain in as much details as possible. But as I said, uh, unmute yourself and ask me questions at any point of time without hesitation. So what is chemotaxis? The definition of chemotaxis is motile response to chemical gradient. So there are certain organisms which have this ability that if you put them in an environment which has a concentration gradient of certain chemical, the chemical to which they are sensitive, these organisms show an ability to follow the gradient and move along its direction. So most of the time here in this talk, I will mainly focus on certain bacteria which show this chemotaxis motion, although there are also other type of organisms, some other eukaryotic cells which also show chemotaxis. But for this talk, I will only consider bacteria, especially E. coli bacteria. So this is a cartoon where we have just shown, so there is, let's say, certain container inside which you have some buffer solution, and there are some E. coli cells which are moving, which are swimming in the solution. Then you take a capillary and fill it with certain chemical which attracts the bacteria. That chemical could be some nutrient, some food for the bacteria, or it could just be in general some attractant which the bacteria doesn't want to eat or metabolize, but they still get attracted to them anyway. So if you fill a capillary with some such chemical and dip it in the buffer, and immediately or uh, soon, the bacterial cells tend to swarm around the capillary end. Similarly, if you place a capillary with some repellent, which is some chemical which the bacteria doesn't like, for example, it could be some kind of a toxic substance, then the cells show some tendency to go away from the attractant, go away from the capillary. So this motion is called chemotaxis. You first everything, all the cells were uniformly swimming around in the buffer solution. Then you created a gradient by dipping a capillary full of certain chemical. And accordingly, the cell is responding by moving away or towards it. For E. coli cell, this motion happens via run and tumble motion. What is the run tumble motion? By run, I mean straight along a roughly straight line, the cell swings. Okay. And by tumble, we mean that the cell doesn't go anywhere, but it stays at a point and undergo random rotation about its own body. So tumble actually gives the cell an opportunity to reorient its motion. When it is in the run mode, then it is moving along a straight line. And then after some time it will tumble, and then it will choose a new direction and run again in that direction. The E. coli cell, this is a cartoon I have shown. The E. coli cell has certain flagella associated with it. By flagella, you can visualize a flagella by some string-like object, just like I have shown in this cartoon. So each cell contains about 10 flagella, and each flagellum, its string, is connected to the cell body via a tiny motor, which is called a flagellar motor. And these motors can rotate in the counterclockwise or clockwise direction. When all the motors are rotating in the counterclockwise direction, then the flagella forms a very coherent bundle, like shown here. I am assuming you can see my cursor. If not, tell me. So the flagella, you cannot see the cursor?
I can see the cursor. I wanted to
Good,
share
good.
it.
Okay, thank you. So this flagella, they form a nice helical bundle, and this bundle creates a net force on the cell, and the cell swims smoothly. It runs. On the other hand, when some of the motors are rotating in the clockwise direction, then the bundle gets dispersed. Now you can see here, the bundles are spreading in different directions, so they cannot exert any net force on the cell. So therefore, then the cell tumbles. So this is a typical trajectory. It starts from here, then it runs along this direction, tumbles, changes its run direction, runs in the new direction, tumbles again, runs in another direction for some time, tumbles again, and so on. And
How does it decide after tumbling which direction to move? Is it random
yes,
or it
yes,
has some mechanism?
it is random. It is generally considered to be random. Although when people have done experiment, they found a very weak forward correlation. So they have found that there is a slightly more tendency to move in the forward direction, that is in the new okay, so let me put it this way. Suppose if I consider it in 2D motion, Petri dish, 2D run tumble motion, then if the two successive run directions are totally random, that means any angle between them is possible between 0 to pi. Okay? And then the average angle would be pi by 2. But so that is 90 degree. But when people actually measured this angle in experiment, they found 68 degree. So which means it is slightly more likely to move in the forward direction than in the backward direction compared to its previous run direction. But generally when people describe it, model it, they assume that it is decided randomly. So this question, particular question that you asked is very pertinent, because then the next question will be, then okay, if it is direction is totally random, then how does it decide which way to go? I just now claimed that they can follow the gradient and so on. But then if it is changing its direction at random, then how does it decide which way to go? That happens by modulating the duration of the run length. If it is moving in the favorable direction, then runs are elongated. If it is moving in the bad direction, then runs are shortened. For example, if along the trajectory the cell finds that food concentration is increasing, then it decides it is a good direction I am heading towards, so keep going. In the opposite scenario, if it finds that okay, as it goes on, the food level is going down and down, then it would quickly tumble and choose a new direction to run again. That new direction is random, but it is still, the bad runs are quickly terminated. So for example, here in the right panel, you can see that this is a few run tumble events I have shown, but you see that runs in there is an attractant gradient, the downward cell wants to go, and you see that whenever the run has a positive downward component, those runs are longish. On the other hand, these ones where the upward component is positive, those runs are shortened. So after few runs, there is a net displacement. On the other hand, in the left panel, when everything was isotropic, after a large number of run and tumble, we don't see much net displacement. So therefore, in presence of a chemical gradient, there is a net drift. So remember, I emphasize this point once more. It is possible for the cell to run in any direction, upward, downward, uphill, downhill, any direction. But the net displacement, net drift comes because certain runs are longer and certain runs are shorter. From that perspective, we get the net displacement. So this is the intracellular reaction network which makes it possible. I just said that okay, runs are elongated, otherwise they are shortened, etc. But how the cell does it? There are certain chain of reaction which are happening inside the cell, and that chain, that reaction network is shown in a simplified form in this diagram. So I don't expect you to remember everything about this diagram, because even when I see such reaction diagram, I tend to get scared. But there are few points which I would like to just emphasize, and we will use it later during this talk. So each cell, they have uh, they have certain, these are called receptors on their membrane. So receptors are protein, and they are transmembrane protein. By transmembrane, I mean half of the receptor is sticking outside the cell and half of it is inside the cell. And these receptors can be in two different, so to speak, conformations, two different states, if you like. These states are called active or inactive. When the receptor is active, then two things happen. In the active conformation, they are able to cause phosphorylation of a certain protein which is called key A, and in the phosphorylated state, this key A is written as key AP. It is not important that you remember all this, but just one thing I want to mention here is that this, after this protein gets phosphorylated by an active receptor, it doesn't keep the phosphate group for itself. It donates the phosphate group to two different protein. One is called key Y, another is called key B. First, let us focus on key Y. What key Y does, it gets phosphorylated, becomes key YP, and then in this phosphorylated form, it binds to the flagellar motor. Remember, I told you about the string-like substance. They are associated to the cell. They are attached to the cell via motors which can rotate clockwise or anti-clockwise direction. In the phosphorylated form, this QIP binds to the flagellar motor and cause them to rotate in the clockwise direction, and the cell tumbles. So when the receptor is active, it helps the cell to tumble. On the other hand, when the receptor is inactive, then this phosphorylation doesn't take place. QI cannot get phosphorylated to QIP. Therefore, flagellar switches, they cannot turn in the clockwise direction, and the tumbling is not possible. The cell keeps running. So, inactive receptors cause long runs, and active receptors quickly terminate those runs and cause the cell to tumble. And now from here, it is not surprising if I now tell you that outside part of the receptor, which was sticking outside the cell, it binds to the chemical molecules in the environment. And there, in that, when that binding happens, then the receptors switch towards inactive conformation. So, just to summarize what I said, when you have a large number of nutrient molecules available, the receptors would bind to them and become inactive. And as a result, the cell will keep running. On the other hand, when you have a very small number of chemical molecules or no chemical molecules available for binding, the receptors will become active and cause the cell to tumble. So therefore, you see, this part of the network allows the cell to read the environment and respond accordingly. If I see a lot of food around me, I will be happy, I'll keep running. If I see there is hardly any food, I will quickly tumble and switch a new direction to run. This is called the sensory part of the diagram, that sensory part of the network, that it senses the input signal and responds. There is another part of the network which is called adaptation. Adaptation generally means, and you know, all of you know the English word adaptation, it generally means adjusting to your new environment. Here also, adaptation is used in a similar spirit. So, this phosphorylated molecule key AP donates its phosphate group to another protein key B, which in the phosphorylated form EBP causes demethylation of the receptor. A receptor has certain methylation level associated with it, and in the phosphorylated form, this protein, which is an enzyme, it is causing the methylation level to come down. So therefore, to just go through again what I just said. Active receptors therefore cause demethylation. In the opposite scenario, when the receptor is inactive, then this phosphorylation doesn't take place, demethylation doesn't happen. Nevertheless, methylation keeps happening at a background as a background process because of another enzyme key R. And in absence of demethylation, therefore, under the action of key R, methylation level keeps going up and up. Not only that, there is a negative feedback type of mechanism associated with it. As I said, active receptors get demethylated, but because of the demethylation, activity goes down. This is the negative feedback I was talking about, that if the receptor is active, it will trigger some reaction which will bring down its activity. Similarly, if the receptor is inactive, then methylation happens, and then it is designed such that when receptor is methylated, then it switches to inactive conformation. So therefore, binding to the outside chemical or high methylation value, either of these factors are responsible for making the receptor inactive, and in the other scenarios, either low concentration of chemical or large rate of demethylation causes the receptor to be active. Okay, so just remember, so therefore now you can see why I call it adaptation, that this negative feedback type of mechanism makes sure that if the activity is too high or too low, it gets restored to its average value. Any question? Okay, just interrupt me whenever you have a question. Just unmute yourself. Okay. So this is some slide which will perhaps be less scary. As physicists, we are more familiar with this type of description. So the two states of the receptor, active and inactive, it is assumed that there is a free energy difference between them. And unsurprisingly, that free energy is a function of L, that is the ligand, the attractant concentration and methylation level. I explained why it should depend on this, and this is the mathematical nature of the dependence. So the
Okay.
receptor.
What is L?
Yes.
What is L? Can you tell me again?
Yeah, aim
What
is
is
the methylation level. Okay, it you can consider it some kind of a dynamical variable because it can increase when it is methylating. For example, here under the action of this KIR enzyme, aim goes up, and under the action of this KBP enzyme, aim goes down. Methylation, demethylation. Okay,
okay?
and
And
L.
L is concentration.
Exactly, L is concentration of the attractant. Any other question?
Okay, thank you, sir. No.
Yeah, I am hearing some voice of a girl. So can you say it again? Okay, if there is no question, then I will go on. Uh, yeah, so these are the two. So effectively, you can think of it now like a two-level problem. So far as the receptor methylation is concerned, active or inactive, and it can switch between these two states. And this is the free energy. And in the case when it is adapted or in equilibrium, so to speak, in steady state, then the average activity is given by this Boltzmann form. Okay, 1 plus e to the f inverse, and this angular brackets are expectation values. So in the uh, this is the adapted activity which will follow Boltzmann distribution depending on this f. Remember, if there is a concentration gradient in the environment, that means this L is a function of space. It can in general be also a function of time. In fact, in this talk, I will consider such a scenario. When methylation levels are going up and down, then M is also changing with time. So therefore, this free energy, as the cell is moving around and methylation level is also changing, this free energy is changing with time. Okay, and in presence of adaptation, this is the average activity. This is the average probability to find the. Can you hear me?
Yes.
Okay,
Yes.
sorry, I thought I lost the network connection. Then sorry. Yeah, so it is the average activity which is given by this Boltzmann form. And these are the things I have already described to you. Methylation dynamics is binding, and these enzyme molecules, they are moving around in the cell cytoplasm. They bind and unbind to the receptor and change their methylation levels. And another thing we should keep in mind is large activity causes the large tumbling probability. Okay. There are few other aspects of this chemotaxis network. So far, what I have described is from the point of view of a single receptor. Okay. Now, there are actually few thousand receptors in an E. coli cell, about 15,000 receptors. So they show some kind of a cooperativity. That means if I am active, I would like to make sure that the neighboring receptors are also in the active state. If I decide to switch from active to inactive state, I would like to influence the neighboring receptors to switch to inactive states also. Okay. So why this is useful? Because then you can imagine that suppose there is some particular receptor, half of it is sticking out, half of it is sticking inside the cell, and the part which is sticking out binds to some chemical molecule, and then it becomes inactive. But then all the other receptors which did not bind to the chemical molecule, but they will also feel that presence, and they will also switch to inactive state. So this gives rise to a signal amplification. Okay, so this is actually a very important trick or mechanism used by E. coli cell to sense even very weak attractant gradient. It is known that if you place E. coli cell in an environment where there is a very weak gradient present and you observe the cell under a microscope, you will see that it still follows the gradient. People were confused for a long time. How does it manage to do it? It is a very weak gradient. How the cell is so sensitive? It is sensitive because of this signal amplification obtained by cooperativity among the chemoreceptors. Okay, another issue I wanted to mention is that the um, robustness of adaptation. So I told you about the enzyme molecules, okay, the methylating and demethylating enzyme molecules. These enzyme molecules are present in the cell at a relatively low concentration, about 100 or 200 enzyme molecules per E. coli cell. Compared to that, I have few thousand, 14,000, 15,000 receptors. And therefore, the methylation level of all these large number of receptors are being controlled by this very small number of enzyme molecules. So then, uh, so this gives rise to two things. One is each receptor may have to wait for a long time before its methylation or demethylation happens. Suppose I am ready to get methylated, but I need to get attention from a methylating enzyme such that I can actually increase my methylation level. But there is 15,000 of people like me and only about 100 or 120 TR molecules. So I have to wait for a long time before I can actually change my methylation level, which means methylation demethylation reactions happen on a very slow time scale. So adaptation does not happen very quickly. If there is a certain, quick change in the environment, quick change in activity, adaptation does not immediately respond. It takes some time. It happens, it is a slow process. Because of this, that very few enzymes, but very large number of receptors. But then you can ask me the question that then how come I am actually able to adapt at all? I am ready to get demethylated. For example, the activity is high and I am ready to get demethylated, but then I am waiting, and absolutely no attention from any enzyme at all. So then how come this works? So to answer this question, people came out with some idea which was also experimentally verified. So one concept here is assistance neighborhood, another is brachiation. What is assistance neighborhood? It tells me that if one enzyme is bound to one receptor, it also has the ability to modify the methylation level of the neighboring receptors. This is called assistance neighborhood, which means it is really not being served one to one. Yes, it is bound to one particular receptor, but it can also influence the methylation level of the neighboring ones. That helps when one agent can serve many customers. Second concept is brachiation. If you look up the dictionary, the English word brachiation, maybe you already know it. It's a common word. Most of you won't even need to look up the dictionary. But brachiation is the kind of motion used by monkeys or primates to move from one tree branch to another. If you remember, then it holds on to one tree branch, then grabs another tree branch and let go of this hand. So this way it moves from one branch to another. Similarly, a bound enzyme is also observed to perform random walk on the receptor array. So enzymes are on the cytoplasm. It takes a long time for a receptor to get bound by an enzyme. But that doesn't mean that the enzyme changes its methylation level and immediately unbinds. No, it doesn't happen like that. The enzyme actually does a random walk, moves from one receptor to another receptor using this kind of a brachiation technique such that it is always attached to one receptor at least. And it performs a random walk on the receptor array, and that is how it can one binding event can give rise to a chain of methylation reaction before the receptor finally unbinds and returns to the cell cytoplasm. Finally, this is the run-tumble motion of the cell. As I said, that the large activity gives rise to large tumbling probability. So here in this plot, I have plotted tumbling rate as a function of activity. And you notice that the y-axis is in the log scale. So it's a very steep function. So activity increases slightly and the tumbling rate is a sensitive function of activity. It changes by a large amount. Okay. So this is the model. If you have any question on the model, you can ask me at this point. Then I will move on to the question that we are addressing. Any questions so far? You can ask me anything that comes to your mind. There is nothing called a
Can
stupid
you
question.
go back to the previous slide? I couldn't see the graph. I was away for a bit.
Okay, so
Can
this,
you?
can you see it now? The bottom
Yes, yes.
left? Huh. So this is the tumbling rate as a function of activity. And what I was mentioning is that the y-axis is plotted in the log scale. So the activity changes by a small amount, but the tumbling rate changes by order of magnitude. So what the point I wanted to emphasize here is that the tumbling rate is a very sensitive function of activity. High activity means large tumbling bias, low activity means small tumbling bias, that is long run. Any other question?
What would be activity in this case? What would be the exact definition?
Oh, so each receptor has these two states, right? Active or inactive. And activity here means the fraction of receptors in the active state. So you see that is a fraction which can go between 0 and 1. So out of all the
Okay,
15,000 receptors, which fraction is present in the active state?
got it.
Okay, good. Any other question? Acha, if you don't have a question and if you are talking among yourselves, please mute the microphone. This is confusing me
So,
again and again. Please don't talk while the microphone is, just mute it, no problem. You feel you are bored, you can feel free to talk among yourselves, but please mute yourself first. Okay, good. So then I would like to come to the question that we address. Um, so what I am interested in, or what I wanted to discuss in this talk, is the case of time varying chemical environment. As we said earlier, that the cell can be placed in an environment where there is a chemical concentration gradient, and then the cell would follow those gradients. But then quite often it can happen that the environment itself changes with time. Few examples are, suppose you have a certain chemical which the cell feels attracted to, but the chemical is not very stable, meaning it degrades. It degrades and turns into another chemical which the cell doesn't find any use of. Maybe it's some kind of a food, nutrient. Maybe it is degrading, or maybe it is in an environment where it is evaporating. So then, while the chemical lasts, the cell has to find it quickly and show this chemotaxis motion. Or another scenario could be, for example, there is a source from which certain chemical is secreting. Maybe it can be some other cell, some other organism. And then the E. coli cells are following the trail. And then the source is moving about, source is moving around. So these are the some natural circumstances under which E. coli can experience a chemical environment which changes with time. And we ask two questions, generally two questions. One is, in the case when the chemical environment is changing periodically, going up and down, oscillating, then what is the long time behavior of the cell? And another scenario we consider is if the chemical environment is changing once, not periodically once, but quickly, like a step, as shown here in this plot. There was one chemical level present, and then suddenly there is a pulse, and the chemical level changes. Then what is the response of the cell in such a scenario? I don't think I will have time to cover this short time response part, but I would like to cover the time periodic environment, at least part of it. Okay, so let us focus on the time periodic attractant environment. There are two types of time periodicity I have considered. One is this type, that there is no spatial variation, there is only temporal variation. So this is something interesting. So far I was talking about spatial gradient, etc. But suppose gradient, there is no gradient. In the whole environment, the attractant level or the chemical level is same. But that level is not static. That level keeps changing with time, periodically, with a frequency omega. What does the cell do in that case? And another periodic environment which I will consider, where I have periodicity both in space and time, and I have this traveling wave type of attractant concentration. So it is not like we are the first time we are doing it. Clearly, chemotaxis is a problem people have been looking at for quite a long time now. And people have actually considered this time periodic environment both theoretically and experimentally. But the new thing that we are doing is that in earlier studies, all the, measurement was focused on time periodic quantities. But what we are doing a DC average. What I mean exactly by that, I will come to it. But these are the two things. Okay, one signal is changing, oscillating in time. You can measure those oscillations. Or you can say that I don't care about the oscillation, I will measure some average quantity and I will average over all times, irrespective of the phase of the oscillation. These are called DC measurement. Okay. And what we are showing that even when you have a time periodic input signal, by performing the DC measurement, you can actually learn a lot of new things about the system which were not known earlier. Okay. So let me first consider the simpler scenario where I have this type of an attractant environment where there is no spatial gradient, but there is temporal oscillation with a frequency omega. Now, people have actually looked at this, exactly this problem earlier, that they found that the activity, they measured the activity of the receptor in experiment, and they found that the activity also shows similar oscillation with the same frequency, but there is a phase lag between the input signal and the activity. Whenever you have one time periodic input and another time periodic output, but with a phase lag, you think of hysteresis. You all know what is hysteresis, that when you change the input in a cycle in a periodic manner, the output does not retrace its path, but output, if you plot input versus output, you trace out a loop. So whenever there is a phase lag, there is a possibility of hysteresis, and we are interested to explore this effect. So this was recently, very recently accepted in this journal, Europhysics journal E. So this is the time series. Okay. So this is what I was talking about, this very clean line that you have, that is the attractant concentration. I have given a periodic attractant concentration, and these different colors correspond to different frequency. And what I have plotted here is the time series of the receptor activity. In my simulation, I just measured the fraction of receptors in active conformation at a time t, and I plot that. And here I have not performed any averaging. So as you can see, that indeed receptor activity also shows the same frequency, because, okay, roughly speaking, you can exactly cannot see that the frequency is same, but you can judge, right? You can see 1, 2, 3 maximum, and for each activity time series also you can see 3 maximum. But as you can see also, the amplitude and the phase lag depends on omega. For different values of omega, I have different activity amplitude and different phase lag. So here I have plotted the activity amplitude and the phase difference as a function of omega, and that is the green plot, okay, green or this line, and the black points I have plotted this quantity 1 plus e to the f inverse. So this is the value of activity expected if Boltzmann distribution was being satisfied. That is, if I have a kind of quasi-equilibrium approximation which remains valid, that even though free energy, etc., these are changing periodically, the activity switching still manages to equilibrate, and then the average activity will indeed be given by this Boltzmann distribution. And then I have plotted the amplitude and phase of this quantity also. Okay, so what are the first things that I want to show? I want to emphasize, that is, first of all, just qualitatively look at it. Activity amplitude increases, reaches some plateau, and then decreases again in the large frequency limit. Phase lag starts from a small value. Initially, it changes slowly, but then it picks up, changes rapidly, and then again in the very large frequency, it kind of saturates to value which is equal to, this is roughly 1.5, right? So I am plotting delta phi by pi. So therefore, it is roughly saturating to the value 3 pi by 2. These are my simulation observations. So now I would like to explain these results. So I have three different time scales. If you remember, one time scale is time scale for adaptation, methylation demethylation time scale. That is a slow time scale which we discussed why. Another time scale is activity switching. The receptors jumps between active and inactive states. What is the time scale associated with that transition? And there is another time scale which I have put externally, that is time scale associated with variation of the input signal, 1 over omega. Now, in the limit when omega is very small, there is temporal periodicity, but it is changing very, very slowly. Then you can imagine that for all value of the input signal, the cell manages to equilibrate, because it is changing so slowly, it is almost like quasi-statically. And then you can imagine that at all times the cell will be adapted. The activity will adapt to the current level of the chemical. And then the receptor activity will follow Boltzmann distribution for all these times. And indeed, if you remember here, you see that the black line, the free energy line, and the simulation observed activity line, they exactly match in this regime. And in fact, long time back, one group had actually calculated the activity amplitude and phase in this limit of very small omega, and they had shown that the amplitude scales linearly with omega and the phase lag takes a value pi over 2. So compare it with my data. I am finding some exponent which is about 0.85. Not exactly linear, but then I also am using a slightly different model, but it's not very far from linear. And the phase difference here, actually we are starting from 0.6. We were not able to go to very small frequency because it is computationally very difficult to get clean data in this regime. But it is at least consistent that we see that it is decreasing, and perhaps it is decreasing towards 0.5. Then, so this is the limit when the time scale is very, very slow. Then when the frequency increases, then finally it becomes faster than the adaptation time scale, because adaptation is the slow one, right? So when the input frequency, we call it in the intermediate range, when the methylation cannot keep up anymore, and then the variation in the free energy is fully controlled by the variation in the ligand, because methylation doesn't change much. Methylation is kind of the input signal is changing quite rapidly, and the methylation demethylation reactions, which are slow process, cannot keep up. So the activity is still given by this, because the activity switching is still happening, keep satisfying this Boltzmann distribution. And if you just look at the form of Ft, let me go back to that form once more. Remember, this was my free energy. Instead of L, now I put in this time periodic value and I calculate which value of L will minimize the free energy. Okay. So, so what is the amplitude? Yeah, the free energy is minimum when sine omega t is minimum. That is, omega t is equal to 3 pi by 2. That is when the sine becomes -1, right? Then the free energy is minimum, and therefore that is when the activity is maximum. Because if you see this form, E1 plus e to the ft, when f is minimum, the activity will be maximum. So when the ligand free energy will be maximum, when the ligand will be maximum, LT, the input signal, is maximum when sine is maximum, that is at omega t is equal to pi by 2. So the activity peak happens at 3 pi by 2 and ligand concentration peaks at pi by 2. So the phase difference is pi. So this is consistent with what we find. So in the intermediate range, you see that it is actually this is 1. Okay, this is close to pi. But then when the activity varies very fast, what, how much time do I have? Okay, it is 48. When the activity varies very fast, then we are in the high frequency regime, and then this Boltzmann approximation is not valid anymore, because I cannot describe it as a quasi-equilibrium process. And then I actually have to solve this particular equation, which is the time evolution equation for activity. And you can actually solve this equation analytically. I'm not going into the details of analysis. It's a simple solution. And this is the form of the solution in the large omega limit. And as you can see, the amplitude, this is given by this, it varies as 1 over omega. Here C1, C2, these are some constants which do not depend on omega. So far as omega dependency is concerned, the amplitude scales as 1 over omega. And what about the phase? I can say that, okay, this average activity will be maximum when this cos omega t is maximum, right? Now the cos omega t will be maximum at 0 and 2 pi. But 0 will not count, because it is, I know that activity is lagging behind LT. So the LT peaks at pi by 2. What is the next peak of activity? That happens at 2 pi. And therefore the phase difference between them is 3 pi by 2, which is consistent with what we find here. Okay. Now, here I have shown the hysteresis loop. Here I have plotted this activity as a function of ligand concentration during one complete cycle for different omega values. And you can see this is a very nice, different hysteresis loops are obtained. Both the loop area as well as the loop orientation, they depend on omega. And you see this red one, which shows a very thin strip. The hysteresis loop area will be very close to 0 here. And the reason is actually very simple, because we argued that for some intermediate value, the phase difference becomes pi. What is the meaning of phase difference pi? They are exactly out of phase. So when the ligand concentration is increasing, activity is decreasing. Immediately, phase difference pi. So when the phase difference pi, then the loop, there is no loop actually. Then for each value of the ligand concentration, there is a unique value of activity. This happens when the phase difference is pi. So then there is no hysteresis. So here I have plotted the loop area as well as loop orientation as a function of omega. And as you can see, as we discussed, the loop area increases first then decreases. This is where it is actually becoming very close to 0 because the phase difference is close to pi, and then it increases again, and finally for very large omega it decreases. And since we actually have analytical solution for activity, we can actually plot the area, loop area, which is this, we can actually calculate the area, and this by performing this integration, and this area finally in the large omega limit, the area has this from 1 over omega, and this actually matches with this slope here. I have not shown it here, but this slope, if you measure, it is actually falling off as 1 over omega. So I think it is 1952. So I will stop here. If you have any question, you can ask me. I think we should keep some time for questions. Question, comment, anything.
Okay, so the first graph, why is there sudden sharp dip in the graph plot?
This one, this slide.
Yes.
Okay, so this is what I mentioned, that the intermediate frequency, when the frequency is around this value, then the phase difference becomes very close to pi. A pi phase difference means one is going up, another is immediately coming down. So maximum of, let's say, maximum of ligand concentration coincides with minimum of activity. That is the pi phase difference. So in this case, activity becomes a single valued function of attractant concentration. Then when I plot activity versus attractant, I don't trace out a loop, I trace out a line. I mean, if you remember why there is hysteresis, because there is this single valuedness is missing, right? There is a loop. For each given value of ligand concentration, there are two solutions of activity are available. But when phase difference becomes pi, those two solutions merge. That is why there is no loop here. So the loop area vanishes. Any other question? Okay, so there is a question in the chat. How have you done it? No, we actually use Monte Carlo simulation mainly, not MD, Monte Carlo simulation. Okay, if there is no other question, I thank you for your attention and I will sign off. Thank you very much.
But one question, will we get the PPT for this talk?
Sorry?
The PPT that you have presented here, will we get that in the WhatsApp group?
In the WhatsApp group, this talk is being recorded, no?
Okay, so recorded lecture will be given.
Yeah.
Okay. Thank you
Thank
for
you.
providing with us such a interesting topic.
Okay, thank you very much. Bye.
Ma'am, I would like you to wait for 5 minutes.
Yeah,
Thank you so
5
much, ma'am,
minutes.
for that.
What happened?
Yes, ma'am. We need to thank you. Thank you so much, ma'am, for that highly insightful and engaging talk. It was truly an enlightening session that gave us a deeper understanding of bacterial chemotaxis and time-varying environments. Before we move on, I would like to take a brief moment to apologize to you, ma'am, for not being able to provide the formal introduction at the beginning of the section and for the initial lack of response due to technical difficulties on our side. We deeply regret the inconvenience and sincerely thank you for your patience and understanding. And now, As we come to the end of today's session, I would like to welcome Ms. Amana Fatima to deliver the vote of thanks and formally conclude the event.
Excuse me, am I audible?
Yes.
Yes.
Am I audible?
Yes, it's audible.
Yes.
Hello, ma'am. Am I audible? Hello?
Yes, you're audible.
Yes.
Yeah. Ma'am, I guess there is one question in the chat. There's a technical issue with Amana, so she'll be joining soon. Maybe you could address that question.
Sorry, what is the question?
The question is if bacteria respond to gradients of non-chemical stimuli, example light, sound, magnetic fields, does that redefine the boundaries of chemotaxis itself?
No, chemotaxis is specifically for the case when it is responding to chemical gradient. But there are other types of gradient. For example, magnetic field, it is called magnetotaxis. When it is responding to certain light intensity, it is called phototaxis. So those are also possible processes, but they possibly follow different types of reaction network. Okay, so that will not be covered within the realm of chemotaxis mechanism.
Okay, thank you, ma'am. I guess Amara have some technical issues, so I will conclude the session. So thank you,
Okay,
Dr.
thank you very
Chatterjee,
much.
for your insight on bacterial chemotaxis and especially in the case of bacterial cells. Your guidance will surely help us explore new avenues in physics. We sincerely thank our HOD, Dr. Vairi Shivaji Reddy, and our faculty coordinator, Dr. Natishan Yogesh, for greatly supporting Bhauthiki in organizing this event. We thank our audience for showing great enthusiasm to join our event, even though an online medium. So your presence meant a lot to us. So thank you, everyone.